Well, good morning, everyone. So I'd like to welcome you to today's installment of the Royal Tyrrell Museum Speaker Series, which is sponsored by the Royal Tyrrell Museum Cooperating Society. And this week we have a guest who's here all week working in collections on research projects, thanks to some additional support from the Cooperating Society. Our work takes place through the provinces known as Alberta. We gratefully recognize and honor Indigenous peoples' long-standing connections to these lands. And today's speaker is Dr. Ian Wilenzik, who studies the evolutionary history and biogeography of squamates, which is a group that includes snakes and lizards. So Ian earned his bachelor's degree in biology from the College of William and Mary in Williamsburg, Virginia, and then went on to do a PhD at George Washington University in Washington, D.C. And he is still at that university as a visiting assistant professor, balancing his time between teaching introductory classes, including vascular plants, and doing research on squamates, and particularly morphological changes in snakes across extinction boundaries. So today, Ian's going to be talking about some of his work on the biogeography of lizards and snakes. So Ian?
All right, thank you all so much for coming out with a very warm welcome interpersonally and a bit of a cold weather welcome. All right. All right, so let's talk about some general background, because there were some words in the introduction that I used that we'll make sure that we define so we're all on the same page here. So what is biogeography and why lizards? So biogeography is the study of the distribution of species across geographic space, so across the planet, and geologic time, so all the way back to the beginning of the world. Fossils are really, really important for the study of biogeography. Basically, we can kind of thank fossils for realizing that the continents aren't where we are, aren't where they are today, that they actually have moved over time. So this is one of the first ones that was looking at some, specific species and realizing that their ranges kind of made the continents fit together like a puzzle piece into one. So we're going to learn some terms here. And we're going to use these terms throughout the presentation. So the first term we're going to learn is the name for the continents. So when I'm talking about Eurasia, these are the northern continents, so North America, Europe, and Asia. When I'm talking about Gondwana, I'm talking about the southern continents, so South America, Africa, Australia, Antarctica, and India actually as well as a subcontinent. And when I'm saying Pangea, we're talking about the whole, every single continent all into one. Next one we'll say is area. So area is a specific single unit. So for instance, if we want to look at North America, right here, North America would be our area. If we want to kind of look at more than one, we call that a range. So a range can be one, so North America, or it can be a combination of ranges, so North America plus Europe. The reason we do this is because what I want to do in my research, and often one of the results of biogeographic analyses, is the ancestral range estimation. Basically, where did these species come from? So for instance, if we found that, through our analysis, this group was originated in kind of the area of North America and Europe, then we'd say it has an ancestral range of North America and Europe. For example, this is one that was done with camels, or members of the family Camelidae. So they are found in Africa, Asia, and South America. However, when you do the analysis and include some fossil taxa in North America, we find that the ancestral range is actually in North America, where then it's spread to Asia and Africa and to South America. Next, the last one we'll talk about today is dispersal. So dispersal is basically the movement of one species from an area to another area. Think migration. So if you're going from North America to Europe, that's a dispersal event. All right, so now we've kind of got on the same page with biogeography. Why are fossils important? Well, one thing is that extant data is often not enough to make a lot of these ancestral range estimations accurately. There are some instances where you can. So for instance, this one, they're all in the orange, all throughout the tree, so originated in the orange. Excellent. What if we have no modern representatives like we have in this light blue? Well, you can't use extant data for that. You have some instances where it is ambiguous where it originated, because some in equal parts of pink and yellow. But if you look at the fossils, the earliest fossil is in pink, meaning that it's likely originated in the pink area. And then you can have a really complex one where there's, one representative in the purple, but a long, long history of the fossil record showing that it's actually in a different area. So in general, no, extant data is often not enough to make these large conclusions. And that's why we have to bring in fossils, which is, of course, why I use the best group out there, squamata. Also known as the squamates, so I'm going to use squamates pretty much throughout the rest. When I'm talking about that, I'm talking about lizards, snakes, and these guys right here, which are amphisbaenians. They're like worm lizards. They're really cool. They are a very, very, very diverse group. They contain about 11,000 species with a fossil record of about 1,000. So for context, that is more than mammals, turtles, and crocodilians combined. So there are a lot of squamates. They're also really cool for other reasons. So they lose their limbs at basically every opportunity. So this is the only snake up here. The rest of them are just forms of limbless lizards. They also very well known for venom. It's not just snakes. There are a lot of other organisms within squamata that have venom. And they're also known for these incredibly flexible skulls in snakes. So all of these are really cool. The other thing is that they are everywhere. It's kind of hard to believe in negative 20 degree Fahrenheit weather, but squamates make it all the way up here into Alberta as well. So they're everywhere. And they're also really, really old. So this red star down here is the earliest kind of early squamate, about the middle Triassic, with the first crown or first modern-like squamate happening in the Jurassic. So they are old, old, old. Some of the groups that we'll talk about kind of interspersed. One of them are the mosasaurs, which you will see plenty of examples out in the museum. These are giant marine lizards, carnivorous, basically the top predator of their time in the ocean. At the same time, you've got the polyglyphanodontians, which are still large, not quite as large, but large herbivorous lizards that basically are the only ones you can really find in a lot of bone beds in the late Jurassic or the late Cretaceous up north. And then one lesser known, but still kind of interesting, are the paramycelodids, which are small little insect-eating lizards of the Jurassic. So, because of their broad geographic range, so they're everywhere, because they're so old, this is a perfect group to start doing some of these large-scale biogeographic analyses. And that's what I did for my thesis. So my basic research questions were, how and when did squamates actually achieve this global distribution? How can we integrate fossils into some of these larger trees and larger analyses? And how does the continent's movement actually affect a lot of these? So we're going to kind of go through three parts to this talk. The first part, the basics of squamate biogeography. Then we'll go a little more complex, a little more in depth, and then look at one specific event where something weird is happening. All right, so let's talk about the basics. So if you want to read more about this, this was published in PeerJ, so if you want to see everything I did, feel free to look further in that one. All right, so this is a diagram showing all of the late Jurassic and early Cretaceous squamates. So you can see pretty large Eurasian range, and that's going to be kind of a theme throughout the rest of the talk. So looking at that, I started to ask myself, where did they come from? So again, big question, where did squamates come from? And then we also wanted to see, are there any places where they're restricted during the Mesozoic, again, the Jurassic to the Cretaceous in this instance. All right, in order to do a biogeographic analysis, you need three things. So I made a nice little formula here. And I'll break down each one as I build up kind of how I did this. So the first thing you really need is a phylogeny. You need your biogeographic areas, so we talked about that before. those little single units of interest, and you need a mathematical model of some kind. Put those together, and you get a biogeographic analysis, and you can make conclusions based off of it. So, phylogeny, I used that word before. For those of you who don't know, this is a really kind of a fancy family tree. And it shows how different species are related to each other. So if they're closer on the tree, like these guys, they're more related to things that are further away from each other. This is the tree I used in my first chapter. So this is a massive, massive, massive tree. This is only a subsampling of it. It's about 5,500 species total. It was published by a member of our old lab. So this was our phylogeny that we used. So pretty big, one of the largest, well, I won't spoil it too much, but yeah. Then we look at our biogeographic areas. We basically mapped a bunch of shapefiles together to make these 9 areas. And then we use that to automatically assign each squamate to a specific area. Now, 9 areas was completely unwieldy and really hard to talk about. So what we did instead was when we reported our results, we kind of condensed it down into 4 different areas. 3 of them you've already heard before: Eurasia, Gondwana, Pangaea. And we also made a separate one we called Northern Pangaea, because a lot of the reconstructions were happening kind of in this northern portion rather than the southern. So we just kind of lumped Africa into Eurasia right there, and we called that Northern Pangaea. All right. So let's talk about the model I used. I'll break it down it's not too complicated once you break it down. So it's called dispersal, extinction, cladogenesis, and jump dispersal, or DEC-J. We're just going to refer to as DEC-J. So let's say you want to go from area A to area B. It's pretty easy, right? You just step over it. The model doesn't allow that, because that's not actually how a lot of these dispersals occur. What the model does is first you can't do that. First you disperse into area B to make a range of AB. So again, you've gone from A to AB. Then you go extinct in that original range. So extinction, AB to B. So you can't go directly from A to B, you have to go through an intermediate. So that's the DEC part. Then there's J, which is jump dispersal. Basically, it allows you to move to a different area that wasn't in that range right here. This is to account for things like large oceanic dispersals. So if, an iguana that rafts on, some log across the ocean, that will account for it. We do know these happen too, so it's good to include them, in my opinion. All right, so again, I blathered all around fossil data, and I had no fossil data so far. So what we did was we basically used fossils to kind of determine where certain groups were at the time. And so we did that to basically force the model to include those areas in their ancestral range estimation. So for instance, if we found an early fossil snake in Morocco, then the model would force the origin of snakes to include Africa in its origin. It can include other things, but it would force Africa to be there. So these were some of the dots. These were each of the nodes that we used the fossils to constrain. I won't go into it because it's not that interesting, but yeah, so that's what we did. Because again, we have our phylogeny, we have our areas, we have our model. This is what we get. And it's a little unwieldy, so we're going to break it down to smaller chunks. So the first thing is squamata. Very broad range. This is pretty typical of DEC-J when you go really far back. They get very broad reconstructions. However, on the next step, we get something interesting, which is a Eurasian distribution. So again, this northern continent. So we know that squamates at this time were pretty restricted to the north. This continues on pretty much from the early Jurassic all the way until the early to late Cretaceous, where we get our first dispersal, or the early Cretaceous, where we get our first dispersal into Africa in skinks. Followed by way more in the late Cretaceous, specifically in, I think this is, sorry, let's see. Yeah, the snakes, iguanins, or geckos, and some other larger lizard taxa. So, what are our broad trends? Eurasia, basically, for the vast majority of squamates' evolution. So they live up north for the entire time. Something happens in the early Cretaceous that causes them to start to move south for the first time, and then by the late Cretaceous, there's a mass migration from Eurasia down to Gondwana. So again, Eurasia, Europe, Asia specifically, are the epicenter of squamate diversity for a very long time, and there is a mass migration south, which basically sets up the modern distribution. So the modern distribution of squamates is achieved basically at the end of the Cretaceous. All right, so that was the simple view. Let's get a little more complex. So in order to kind of set this stage, oh yeah, and if you want to read the more complex one, it was published here. We even got like a nice little cover art for ours, which is a lot of fun. All right, let's talk about a little story. All right, transport yourself back to the Triassic, and we have found these species. We call them shapeosaurs. You got the star, the circle, the triangle, and the square. You're a diligent scientist. First thing you do, you make a little phylogeny. So we know this is how they're related. So now we can tell a story with this. Originally, the population was here in Africa with the star. It then spread to the Arabian Peninsula with the circle. It then spread from the Arabian Peninsula to India, the triangle, before dispersing into China up here with the square. There's a problem with the story that I've just told you. This is the modern continents. That's not what they looked like back in the Triassic. This is what the continents looked back in the Triassic. So if we map that same story onto the Triassic continents, we can find that this is not a very likely event, because India's all the way down here, and this would require a massive dispersal across basically thousands of miles of ocean. This presents a problem because a lot of biogeographic analyses do not include any measure of tectonics or the way the continents move in their models. So again, no information of how tectonics work. So how do we take this tectonic plates fossil data and then get to something similar to what we've discussed previously? So my advisor, Alex Pyron, and I worked on this. So the first thing, remember, the first part of our formula is phylogeny. So what we did was we took a bunch of, we call them total evidence, which is basically, your DNA and some sort of, measure of structures, and you put them together and it makes a tree. So remember some of the fossil taxa that you're going to hear about again: mosasaurs, the large marine, polyglophanodontines, the medium-sized herbivores, and the smaller insect-eating lizards. These were the ones we used. So the first one was based off a matrix by someone named Conrad. We called it Con. The Gautier matrix we called Gao, and the Samoas matrix we called Sim. And those will be used throughout. So what are our areas? So the areas and model kind of go hand in hand for this one. We based all of this off of the work done by Michael Landis. He's a brilliant comparative methods guy, so we kind of borrowed a lot from him. Me and my advisor, Dr. Alex Pyron, adjusted his original model for the use of ancestral range estimation. So in the original model that we've adapted, there were 25 areas preset. So there's no guessing on it. You just use the models that were, or the areas that were set. Very easy. There are also 26 time slices. So basically, they called them epochs, which took different slices. And we'll say I'll talk about what they did with that in a second. All right, so the model itself. For each of those time slices, it basically says how close different areas are to each other. If they are short distance, if they're close by to each other, connected by a black line on this diagram, it means there's a short dispersal, very likely to happen. If there is a white line, so instance southern South America and southern Africa here, it's medium, which means it could happen. It's less likely than short, but still it still is possible to happen, followed by long distance. Basically, if anything's not connected by a line here. Long distance, least likely to happen, very infrequent. These were then used to create what is called a communicating class, which is a fancy word for saying basically things that are closely connected to each other. So in this diagram up here, the early Cretaceous, there are 8 communicating classes: North America, Europe, Asia, Africa, South America, India and Madagascar, and Antarctica and Australia. So each one of those time slices I talked about basically accounts for, there we go, changing communicating classes. So each time slice has a different communicating class across it. It can then track how changes occur, accounting for how the tectonics or how the continent's orientation have changed. So again, we're going to ask kind of the same question, where are squamates coming from? But this time we're asking, how do plate tectonics actually affect that original analysis? Then we can also ask similar questions of how did they spread out before the KPG mass extinction, the one that ends the Cretaceous. So a little bit of a primer so you can read some of the graphs I have up. Basically, it comes out as a little pie chart. The larger slice of the pie, the more likely that area is. You'll see some in gray. Gray just basically summarizes the other ones. So the more gray there is, the less certain your results are. We say that switching when there's a majority one color to another is a dispersal event. And if they're about equal, we count that as a combined range. All right, so let's look at con first. So the origin of squamates all the way up here, this kind of tan color is Europe. So squamates in this originate in Europe and then disperse into Northeast Asia, where they stay pretty much throughout the late Jurassic up until the early Cretaceous. It isn't until the early late Cretaceous that we start seeing our first movement south into Gondwana, specifically in snakes and iguanas. All right. Fossil groups, so again, we're talking about those. The paramesolodids, Europe, pretty consistently. The mosasaurs, also Europe. And the polyglyphanodontians, Northeast Asia. So let's look at the Gautier one next. Squamates actually have a bit broader range that includes Northeast Asia and Europe. But after that, pretty much only in Northeast Asia, where we see a no dispersal event happening in the late Cretaceous in iguanas and in snakes. It's going to be a repeated pattern. Fossil groups, the paramesolodids are joint, so more broad range of Northeast Asia and Asia. The mosasaurs, Europe again, and the polyglyphanodontians, also Northeast Asia. And the last one has the most kind of early groups, but not as many later groups, so it's harder to make large conclusions about that. But we can look at the early groups. Basically, squamates originate in Europe. There's really no other question based on this one. And they stay in Europe for a very long time. The first expansion into Northeast Asia occurs in the middle Jurassic, so a little bit later than the other models suggest. And the first dispersals occur in the late early Cretaceous and the early late Cretaceous in snakes and iguanas. Fossil clades here, there weren't enough paramesolodids to make a conclusion. However, polyglyphanodontians have a broad Northeast Asia and European origin, with mosasaurs being in Europe. So, what does that mean? So basically, that Northern Europe plus Northeast Asia communicating class right there is basically consistently reconstructed as that origin of squamates. So specifically to Europe for most of them as well. Europe is right there. And it displays pretty much only Laurasian ranges up until the late Cretaceous, that boundary. That's when squamates move south. Again, so right here we can see the some examples of some of those dispersals south. This is extremely consistent with our results from the other group. So again, both suggest a Laurasian origin for most of the Mesozoic up until the late Cretaceous, where they start moving south. The one thing that is different from this result is that it pretty much suggests that squamates originated in Europe rather than a broad kind of Eurasian-Laurasian range. The fossils are exceedingly consistent, though, which is very interesting. Mosasaurs pretty much only originate in Europe. There is no real debate about that one. Polyglophanodontines also originate mostly in Northeast Asia, and the permacelotids are between Europe and Northeast Asia as well. So again, one of the big things is that this is a pretty powerful new method to reconstruct biogeography when you are going very deep into the fossil record and have very broad ranges. So this is a really exciting new one that I'd be very happy to see more people use, so yay. It does really show a European origin for these groups. So all of these originated from a single population, likely in Europe. Northeast Asia is kind of a refuge for a lot of these species for most of the Mesozoic, up until they, start to spread out from there. And fossil clades are consistent across the tree. So the next thing we're going to talk about is a weird little event happening that is not super studied, and that's kind of going to be my next step of my academic career. All right, so we looked at global biogeography. We've looked about it everywhere. Now we're going to examine one specific area to see what's happening. We're going to look all the way up here. So Eurasia, again, just a quick reminder, Europe, Asia, and North America, in the late Cretaceous looked a little different than it does now. For one, they were actually connected by a land bridge. So this is kind of analogous to the land bridge that is hypothesized to have allowed humans to cross over from Asia into North America. So, based on the geological record, we know pretty confidently that Eurasia, specifically from Northeast Asia, was connected to North America there. So, we remember the polyglophanodontines. One of my chapters was to redo some of their phylogeny. So to add a bunch of species, see how they're related to each other. Just because I love biogeography, I decided to throw it into a biogeographic analysis just to see what comes up. And the results at first were kind of boring. Northeast Asia, kind of what I expected, consistent with the other results I had. I started to look a little bit deeper though. First thing I noticed was that there was only one instance of dispersal in the group from Northeast Asia into North America. Pretty typical, there's only really one that I added in. So I started looking kind of outside of that at some of its closely related groups, and I found that, again, there was another single instance of dispersal from Northeast Asia to North America. About the same time, too. So very weird that it's not happening really any other time. It's really only happening there. I did notice that, okay, there's no movement back from North America to Asia. You kind of expect if a land bridge to connect two continents, you might expect some exchange, but no, this is just one direction. But, that's only two examples on one phylogeny. What are you going to do? So I kind of just forgot about it for a bit. Then I was talking to my friend Jenny Brennan. She's actually visited the museum about 2 years ago. She works on these guys up here, which are the dome-headed dinosaurs called the pachycephalosaurs. They also have a very similar range of Eurasia, so Asia and North America. And so she's working on the phylogeny as well. And I was like, you know what? Let me throw that into my biogeographic machine. Let's see what happens when it comes out. What I noticed, again, 2 instances of Asia to North American dispersal around about the same time. All right. Kind of cool, but still no migrations back from Asia to North America. So I started to look into the literature at the time to see what other species there might be that are moving across this land bridge. There weren't a whole lot because there isn't a lot of quantitative biogeography at the time. So hopefully, and I'll talk about that, hopefully that's kind of the next step. I found this paper on tyrannosaurids. So again, family of T-Rex. Put the T-Rex in your paper, you get into nature kind of thing. So there are 2 independent dispersals of tyrannosaurids from Asia into North America, here and here. We actually see our first instance of the reverse of that, from North America to Asia, but it is right here in the very late Cretaceous. So in the earlier late Cretaceous, pretty much they're only going one direction, from Asia to North America. So that's a weirdly consistent pattern of that early late Cretaceous movement of Asia into North America. So again, just based off of brief literature review of kind of the other places people have talked about it, they've suggested similar patterns in a lot of diverse groups, including other dinosaurs, so ceratopsians, the frilled dinosaurs, dromaeosaurs, include velociraptors, and also turtles, some group of amphibians, angiosperms, the flowering plants. So this pattern seems to be pretty repeated of starting in Asia and ending in North America. This suggests that there is a massively asymmetrical migration pattern happening here when these two continents are connected. So is that consistent with other times that continents have connected? So the most famous probably is called GABI, which is the Great American Biotic Interchange. This is when North America and South America collided, made a land bridge called the Isthmus of Panama. Now, while it isn't a completely symmetrical migration, a lot of species moved either from south to north or north to south. Varying degrees of success, but they were still doing it. So why is this Asian-North American land bridge not acting like GABI? What is different between them? Now, I don't have an answer, unfortunately, but I do have some hypotheses that need testing. So the first one is I've called the extinction hypothesis. Something happened in North America at the time to greatly decrease the biodiversity there that basically allowed Asian taxa to flow in and occupy some of those ecological niches left open. It's a bit challenging to test. There are some cursory support for this, though. Right around that time is when the Western Interior Seaway opened up and basically split North America into two. Anytime you get a large amount of change geographically, it tends to correspond with some local extinctions around there. Again, you're changing the way the environment is. Species that are adapted to a specific environment might go extinct there. So that's some support, but still not enough to make a conclusion about it. What I would actually need is some measure of biodiversity at the time from the early Cretaceous into the late Cretaceous. If we're seeing a change in biodiversity there, a large extinction event maybe in this localized region, that would provide some evidence. So that's number one. Number two, I called the takeover hypothesis. Basically, the flood of Asian taxa into North America was based on the fact that the Asian taxa, for some reason, were better adapted and could outcompete the North American taxa that were there previously. This one is by far the most challenging kind of hypothesis to test. Competition in the fossil record is difficult to test. One potential avenue I could take that I talked about with one of my friends who did this in hominids would be look for Hutchinson's rule, which basically suggests that when there's competition between two species, one of them has to give, and they change their morphology by about 1.3 times. So this happens in humans. We actually changed because we were having trouble outcompeting Neanderthals at the time. So if we can find some early, late Cretaceous taxa in North America, test their morphology, see how much they're changing, and if they're changing by about 1.3, that suggests increased competition. Hypothesis 3 is kind of a combination that North American taxa were decreasing while being outcompeted by taxa from Asia. Probably likely. Again, oftentimes biology likes to live in those like little gray areas in between hypotheses. However, again, future work is required for me. I like having work to do, so that's good. And it's kind of on my long list of things to do, including teaching non-majors biology, which is fun. So again, I'm kind of doing this off of not a huge sample size of migration events. So I want to look at a lot of those kind of the angiosperm dispersal, which is just talked about but not actually quantitatively looked at. So I do want to see how these organisms are actually spreading in a larger sense. And then start to test some of those hypotheses. See if there's a good candidate species to see if there's any changes occurring. All right, well, some conclusions from my talk. One, propose some advanced methods to help kind of reconstruct where these old, diverse, and dispersed lineages come from, both by adding fossils as kind of, geographic information, and then also adding tectonic information into the model itself. We found the potential for an asymmetric migration event, which the reasons for it are still unknown and should be studied further, which is an excellent source of future research, and hopefully give a more complete view to squamates and how they got to where they are today. All right, and with that, I've got tons of people here to thank, basically all of my collaborators, any curator I've ever worked with to help make these projects a thing, some of my really close work colleagues, and of course, family. So thank you so much for attending, and feel free to ask questions.
So I was just wondering, I mean, it looks like you've got data from all over the place. I was wondering if there was any issue, like I know in sort of macrovertebrate paleontology, there's an issue with a lot of like research and collection historically going on in North America and Europe and stuff. Is that something you had to deal with?
Yes. So one thing that we're pretty confident on is that a lot of squamates did originate in Northeast Asia. There are some other taxa happening at the same time that show a similar pattern. So that is definitely something we're a little more confident in. However, a lot of these, specifically Africa, you are correct, is undersampled for many, many, many reasons. So hopefully further sampling there will help either support the idea that these are mostly Laurasian taxa, or perhaps we'll find new examples of Gondwanan taxa that will expand our view of them. But yeah, absolutely, that is something to consider. And I did talk about it in both of the papers, that it's an important place for future development. Yeah, absolutely. Thank you.
In the museum towards the tail end of the display, there's a brief mention of what has been referred to elsewhere in popular literature, not the scientific, but the Great North American Mammalian Extinction Event. Does that lower scale type of event provide an opportunity to look at the asymmetric migrations that you referred to? I mean, the later Cretaceous event might have been actually really big. The more recent one's kind of small, but not if you lived here and you were a camel or a mammoth.
Yeah, absolutely 100%. In fact, probably some of those smaller events that are better sampled, more modern, would be a great place to test a lot of the smaller methods I would propose. So yes, absolutely, those would be extremely helpful and good sources for future research.
I'm just wondering if you have any explanation for why the squamate suddenly went south in the early Cretaceous. Were there some sort of reptile analog doing the same thing, same ecological niches, and went extinct?
That is a great question. The answer is weirdly no. There really wasn't anything there to block them from spreading. Their close kind of cousin are called the rhynchocephalians. The Tuatara are the only extant member or living member of that. They spread south at about the same time. So it's not like there's out competing. They both kind of spread south. The reason is unknown. Definitely an area for future research, but it's weird that it happens at the same time in all groups, but there just doesn't seem to be a reason for it that we know of quite yet. But yeah, that's definitely an area of future research. So Teresa says, do you think new discoveries from mid-Cretaceous formations in Western North America, such as the Wayon Formation, could help shed light on these migration events? Absolutely. That's basically what we would need to look at for those, to look at extinction events happening in mid-Cretaceous North America. So yes, absolutely. It's actually a good thing to lock away to look for myself.
I don't know if I might have missed this when in your talk, but were there any trends in dispersal patterns among like the different parts of the different taxa in the group? Like, I don't know, like lizards versus snakes, or within different groups of lizards or something like that?
Absolutely, yeah. So snakes are actually weird. They have one of the higher rates of being in different places, different internal relationships than pretty much any other one. So the snakes were a bit weird. They were inconsistent, and so I think that further study on those would definitely help elucidate that a little bit more. But yeah, there definitely were different patterns, although for the most part, a lot of them basically spread to a specific area and then stayed there for a very long time before spreading out further. And especially as the continents get more and more spread out, that's pretty common in some of those larger scale biogeographic events. As the continents get further apart and less connected, they kind of just stay put. Yeah.
Is there a group that did the best?
Probably snakes.
Yeah.
Again, one of the only squamates that makes it very far up north here are some of the, I think there are 3 species of snakes that live up here. I looked this up because I wanted to know. Yeah, and lizards do not get this far. There's some in southern Alberta, but none get really far up north, except for snakes. So the question was, since a lot of squamates eat insects, so like the paramycelodids as well, are they just following the dispersal of insects? And so one of the things that kind of lives in the back of my head is there's something called the Cretaceous terrestrial revolution. Basically around the time that angiosperms or flowering plants really explode in diversity comes a huge explosion of diversity in other smaller animals. So insects radiate, squamates radiate, small mammals radiate. And so that might be one of the contributing factors, is this expansion of insect populations might have driven them further south. However, I don't know as much about insect biogeography, and I know that there aren't a whole lot of insect fossils to go off of to help kind of corroborate that. However, that is definitely something I'd want to look into to see if those patterns are consistent with each other, because that could be one of the explanations as to why squamates are able to move south.
I know lots of like lizard and especially snake fossils are very small and hard to come by. Are there any sites where there's like an especially abundant amount of like lizard fossils?
Yes. Ironically enough, Northeast Asia. So a lot of the really, really, really complete skulls that I have seen come from Northeast Asia. Mosasaurs are kind of cheating by being so big, and the bigger you are, the easier it is to fossilize at least some part of you. They're also in Alberta. I've looked at many of the microvert sites, so small bits of lizards tend to end up in those pretty nicely. But if you want like a nice, complete lizard, Northeast Asia is a great place. Mongolia, China. And then southern United States also has some pretty nice, complete fossils as well for lizards.
You probably mentioned in your presentation, but it's kind of missed it. When there is an unlikely event, like the migration shown from India to China, how is that actually dealt with in your model? It puts it as less likely, which affects maybe the flow of species, but it's still there as a piece of data. How exactly does that affect? I mean, the whole thing about biogeography is it ties in nicely, sort of like the basis for the original continental, one of the arguments for continental drift. Mm-hmm. But when it doesn't actually fit as nicely, it's an anomalous piece of data. So how do you deal with that?
Yeah.
It just...
Oh, excuse me. So I didn't go into that specifically. However, basically there's, I'll just see. It is called a rate dispersal matrix. Basically, it says how likely it is to happen from between 2 different states. With the long dispersal, it is very, very small mathematically. I think it's 0.001. I'm throwing that number out. I don't quite remember, but it's very low. So that means that the model will preferentially select for the dispersals that are more likely. So the medium dispersals or a series of short dispersals.
But it still has to have that data to be able to do that preference. So it kind of hides the one that occurred at a given time in favor of a more plausible pathway that might have taken a little bit longer. So there's a sort of an adjustment in the time frame.
Yeah, I have to think on that one a little bit, but yeah, I will definitely.
Because I guess the question really is: are you afraid sometimes that a mathematical technique may be hiding some sort of really interesting reality from you because you found one in terms of an almost unidirectional migration that's really interesting from Northeast Asia to North America. So I'm just wondering if you're really afraid that somehow there may be a lot of other things in there, gems and jewels that are hidden.
If you're a biogeographer and say, no, I'm not afraid of that, you've done something wrong. So again, it's just kind of your, I don't want to say best guess, because it's an informed estimation. So if there's another bit of information that comes out, I will put it in the matrix, and if it tells me something different, I will go with a different conclusion. But yeah, that is absolutely something that needs to be considered, especially in paleo, because we're dealing with such fragmentary data at the end of the day. But yes, absolutely, that there could absolutely be things hidden, and hopefully further discoveries will help shrink some of our uncertainty in that. But absolutely, 100%.
I have a question for you. Thanks for your talk. Very interesting, very complex topic. It all happened, but we are looking at the fossil record, which is very incomplete and fragmentary, and there's places no one's even looked for fossils. Mm-hmm. So there's obviously some trends that are appearing, but how confident are you with some of these? Like, the fossil record is so incomplete. For example, snakes were around in the late Cretaceous, mm-hmm. but having worked at this museum for almost 50 years, I've never seen a fossil snake vertebra in the latest Cretaceous rocks here in Alberta. I think they occur in the Upper Milk River Formation, but due to various reasons, they're not here, so there's data there that's not available to you as the biogeographer. So I guess the question is, how confident are you in these conclusions you're making, given the incompleteness and fragmentary nature of the fossil record?
So there's a famous quote that was said, all models are wrong, some are useful. And I used to annoy my advisor about that, because he'd give me a paper to read, and he's like, what do you think of it? And I said, well, all models are wrong, some are useful. So we all had to do that before we could talk about it. And what this means in this instance is that technically everything I just presented to you is wrong. We all have to, kind of come to terms with that. But the usefulness is that the reason I believe it's useful is that it's taking the data we do know right now and seeing what conclusions you can make off of that. And so as I said before, if we get new data that shows something different, I would be confident in the fact that the data I used got the results. How confident am I in saying that it's reality? Low, but that's kind of it is a personal perspective on how we do some of these more complex analyses in the fossil record specifically. So I'd say low confidence in the reality, higher confidence in the results, if that makes sense. Okay.
All right. So a good reminder to never bet the ranch on anyone's results.
Nope.
Yeah. All right. So I'd like to thank you, Ian.